Okay, so we're going to talk about photosynthesis, which, generally speaking, is probably harder than cell respiration in terms of, um, at least in terms of HL content. I wouldn't say in terms of SL content, but definitely in terms of HL content, it's harder um, because there's the entire Calvin cycle and things like that that you have to discuss in the photosystems and stuff like that. But yeah, so we're just going to go through photosynthesis and just a few general housekeeping stuff. If you have any questions, keep putting them in chat. I will take a look as I go along. If there's any urgent things, please ping me because I might miss it. Um, but yeah, so the first thing we're going to discuss is wavelengths and photosynthesis. But actually, before we discuss this, let's go back and let's talk about the definition of photosynthesis. So, does everyone have a definition for photosynthesis? Add it in chat right now. I want to see everyone's definition for photosynthesis, just so I know where to pitch this at, I guess. So, I'm going to go back to the photosynthesis slide. What's everyone's definitions looking like? Okay, yeah, so a few things have already come in. So, um, let me just take a look. There's someone said the transfer of, um, not the transfer, sorry, the change from light energy to chemical energy. That's a good point. Someone else said the creation of carbohydrates in the presence of light in plants. Also a good point. Um, Someone said carbon compounds instead of carbohydrates. I think you can write either, but overall, a definition for photosynthesis would be the conversion of light energy to chemical energy in plants in the form of carbohydrates. And someone else, I think, added the fact that it's done in the chloroplast. I think that's a valid point. I think if you add that in, it's also valuable. Um, Let me have a think. Is there anything else that you need to know as a general photosynthesis definition? Um, so I think in terms of proper definition, I would say photosynthesis is the process by which plants convert light energy to chemical energy in the form of glucose. I think that's a sufficient example. So the main three things that you need to mention is it happens in plants, it's from light energy to chemical energy, and in the form of glucose. Whether you choose to or don't choose to add the fact that carbon dioxide is used and water is used and oxygen and is produced as a byproduct, I think is a personal choice really, because the main thing is the conversion of light energy into chemical energy in the form of carbohydrates or glucose, whichever you write. So whether or not you include the fact that it's what do I call it? Whether or not you include the fact that of the reactants and the products that are being used is a personal choice, in my opinion. Um, okay, so yeah. The next thing we're going to talk about is wavelengths and photosynthesis. So, wavelengths are important to discuss because, as I said before, um, photosynthesis is the conversion of light energy into chemical energy, and that light energy comes from, well, light or sunlight. Therefore, light needs to be measured in some way, and we do that through wavelengths. So, violet light or purple light has the shortest wavelength, and red has the longest. And the shorter the wavelength is, the more energy a photon of light has. Now, does everyone know what a photon of light is? Yes, no, maybe. Okay, just as a refresher, a photon is basically a package of light or a unit of light, essentially, or a particle of light. That's the only level you need to know. Whether or not that's completely accurate in terms of physics and chemistry does not matter. In bio, all you need to know is the fact that it is a particle of light, essentially. Now, photons are important because those are what are actually getting they're what make up waves of light, and then that's where we discuss wavelength and so on and so forth. So that's why they're important. But the lesser the wavelength, the more energy each photon has in a wave. Now, chlorophyll has a structure. Chlorophyll, everyone should know chlorophyll. It's a pigment in plants. Chlorophyll has a structure that allows an electron within the molecule of chlorophyll to jump from one energy level up to a higher energy level using energy obtained from absorbing a photon of light. So, as we said, the more energy the shorter the wavelength, the more energy a photon of light has. Now, that energy from the photon of light is absorbed by electrons within chlorophyll that therefore result in the electron moving up to a higher level of energy, or therefore be less stable. A graph that shows which wavelengths are absorbed is an absorption spectrum. So if we look at the absorption spectrum at the bottom, there's chlorophyll A, chlorophyll B, which are two types of chlorophyll, and carotenoid or beta carotene. The absorption spectra basically just shows what wavelengths of light are being absorbed most or least. The y-axis is the amount of light that's being absorbed, and the x-axis is essentially just the wavelength of the light, or an easier way to remember it is the color of light. So anywhere from 350 to 400 essentially is purple, and then anywhere from 700 to 750 is red, and anywhere between that is where the spectrum of light exists. So from 450 so from 350 to 750 is where the visible light exists, essentially. So as you can see in the graph, when we're at the 350 to 400 mark, actually I'm going to say 400 to 450 mark, where it's violet or purple and blue, that's where most of the absorption happens for chlorophyll. And then also at past 600, which is where we go into the orange and red lights, essentially. And then for because the absorption spectra is like this, and you can observe that in the green area there's essentially no activity from no absorption from the chlorophyll. That's why chlorophyll appears as green to us. What else do we need to? Yeah, so the carotenoid is one of many other accessory pigments in plants, but carotenoid is basically one that absorbs more bluish to greenish light. So you can see it in the 450 to 500 range. That's where the absorption spectrum peaks for carotenoid, and then drops off completely near yellow and red. And that's because it does not absorb any yellow or red at all. It only does green and blue, as you can see in the next slide. So again, like I said, the main photosynthetic pigment is chlorophyll. Everyone should know this already, which absorbs mainly red and blue light most effectively. It does not absorb a lot of green light. There is some, but not a lot. Most of it is reflected, which makes chlorophyll appear green to us. And then, although chlorophylls are the main pigments, like I said, there are other accessory pigments, like xanthophyll is one of them, and carotene or beta carotene or carotenoid. There's three different names for it. But essentially, those are the two that were mentioned in your textbook. So those are the two that I mentioned. But as you can see, the chlorophyll bottoms out essentially near the green, and that's where it's reflecting and not absorbing most of the green wavelength light, and therefore you want to. Um, I mean, you can see that that's why chlorophyll appears as green, and that's also why plants slash leaves are green, because they have high quantities of chlorophyll in them as compared to other accessory pigments. And yeah, that's about it for this one. And then we've got absorption and action spectra. So we talked about the absorption spectra just now. There's also the action spectra, which is basically the action that's occurring in relation to the absorption spectra. So in this case, when we're discussing absorption and action spectra in relation to photosynthesis, we're talking about the rate of photosynthesis. So as you can see in the graph, the orange line is the action spectrum, and then there's three other absorption spectra lines, which are chlorophyll A, chlorophyll B, and beta carotene or carotenoid or carotene in general. And again, it's similar to the absorption spectra where it's going through the like different wavelengths in the x-axis, and then the y-axis when you're comparing both absorption and action in the same graph, it refers to both photosynthetic rate and absorption rate of the different lights and so on and so forth. One of the main things that you need to know is how to compare the absorption and action spectra. So you talk about how, because chlorophyll and carotene, both types of chlorophyll and carotene are really high in the 550 to 400, or sorry, 400 nanometers to 550 nanometer wavelength. That's where photosynthesis is at its highest, and then around the middle, the green area, it's low, but it's not zero, and this is because carotene is still absorbing some light at this stage, even though the two chlorophylls are not, and therefore there is some photosynthesis still happening, but not a lot because both the chlorophylls are still bottoming out there. And there's still like, it comes down as the chlorophylls and the carotene all come down, and then it starts increasing again once both the chlorophylls also gain the, I mean not gain, but like start absorbing again, because we've reached into the orange and red wavelength zones essentially. And yeah, so and there's, yeah, also, I think most questions you're going to be asked in terms of absorption and action spectra is you need to know what they are, obviously. So you need to know what an absorption spectrum is, what an action spectrum is, and then you need to know the differences, and you need to know how to infer certain data from it. You'll never be asked to draw absorption or action spectra, at least as far as I know. You'll also never be asked to be like, here are the data points, make the like graph type thing. Mostly you'll be asked to do like inferring. So it'll be like, for example, it can be like a database question where they give you this as the quote unquote data, the graph. They'll give you the graph and they'll be like, okay, explain the relation between the absorptions and the action spectra in terms of this plant. And then you have to explain things like the fact that because the chlorophyll and both types of chlorophyll peak at those certain wavelengths, that means that the action spectra also peaks at those wavelengths. And then because they bottom out at certain wavelengths, that means the action spectrum reduces, which means the rate of photosynthesis reduces. But the main point is that the action spectra shows the rate of photosynthesis, and the absorption spectra show the rate of absorption of light. The reason why they're related is because the light energy that is required in photosynthesis comes from absorption of light. So the more light that's absorbed, the higher the rate of photosynthesis, as we'll discuss in the limiting factors later on. But the higher the light intensity, the more the rate of photosynthesis, which is why these two are related, basically. Okay.
So that's absorption and action spectrum. Are we all good, more or less? Yes, I will go into Calvin cycle. That's later on. I'm going to go through everything in photosynthesis today. So, I'm, yeah. So right now I'm going through all the SL stuff. Because SL doesn't have to do Calvin cycle and stuff like that, I don't think. So, yeah. Are we all good with action and absorption spectrum? If data is given, we should be able to make an action cycle. If data is given, no, you don't need to know how to plot it. The point is, it'll give you. They'll never be like, okay, this is the rate of photosynthesis. You need to be able to draw the absorption spectra for chlorophyll. Like, they won't give you stuff like that. They'll only ask you questions based on, like, what is absorption and action spectra. They don't expect you to know how to plot a perfect graph. That's not a requirement. That's not something they expect you to do. That's not something you should do, really, in my opinion. Okay, so next, limiting factors on the rate of photosynthesis. So there's three main factors that limit the rate of photosynthesis: temperature, light intensity, and carbon dioxide concentration. Is pH a limiting factor or no? It can be, but it's not one of the main limiting factors. The reason I say it can be is because photosynthesis is an enzyme mediated process, which means that anything that's a limiting factor or a factor that affects what's it called? A factor that affects enzymes will be a factor that affects the rate of photosynthesis. But it's not prominent enough for you to write it in an exam. The main three ones that you need to know the effect of is these three: temperature, light intensity, and carbon dioxide concentration. These are the three that are given in your textbook as well. Anything else that you might know is still useful to know, and there will be some effects. There will be effects on the rate of reaction, or sorry, the rate of photosynthesis, but it won't be enough of it for it to be considered a limiting factor prominent enough for you to mention in an exam. Right. So there's temperature, light intensity, and carbon dioxide concentration. So temperature, again, like I said, it is an enzyme mediated process. Enzymes function optimally within a specific temperature range. So whatever that temperature range is for the photosynthetic enzymes will be the optimum temperature range for the rate of photosynthesis as well. So then if that temperature is too low or too high, too low, the enzyme will just stop working. Too high, the enzymes get denatured, which means that they can no longer work as an enzyme. And then the enzyme activity will either reduce or be stopped, which limits the rate of photosynthesis, as we know. And also, if it's too high, it also just damages the plant, which, I mean, is an obvious thing as well. Light intensity. So the rate of photosynthesis increases as light intensity increases up to a certain point where the process will plateau, because there's only so much light the photosystems can absorb. If there's not enough light, it becomes a limiting factor, even if carbon dioxide and temperature are optimal. And this will be because if there's not enough light, there's not enough energy in order for the electron transfer to happen, and therefore there's no the photosynthesis can't go on, basically. And then carbon dioxide concentration. So photosynthesis obviously requires carbon dioxide as one of the ingredients, I guess you can say, to photosynthesis to create glucose, and a lack of carbon dioxide can limit the process. As carbon dioxide concentration will increase, the rate of photosynthesis also increases, but again, it will only be to a certain point because there's only so much carbon dioxide the plant can absorb at one point. Do we all understand limiting factors? Okay. I think that's everything for SL. Okay, no, there's more. You need to know, I've not put this in the slides because I forgot to put it in the slides, but you need to know, well, chromatography, yes, but chromatography I'm not going through today. I said this before as well. I'm going to go through it together with other experimental things because I think I want to do a session where I go through every single experiment that you need to know, and then we'll just go through it all together, if that makes sense. But you also, everyone needs to know the word equation for photosynthesis. So everyone write down the word equation for photosynthesis in the chat, which you should all know by now, but I'm gonna ask anyway. Please spoiler your answers, so add 2 bars on either side of your like yeah, while everyone writes down the equation for photosynthesis. Yes, everyone's pretty much right so far. I think in the textbook you can write light as a reactant as well, or you can put it above the arrow, either or. You need to have light, so whichever way you try to say I would put it above the arrow, but I think in the textbook it just says carbon dioxide plus water plus light gives glucose plus oxygen, which I think is fine as well. You can write it like that. It's the same way when you say energy is a product of what's it called, cell respiration. It's the same way. Just that's nice as well, but I don't know if you're required to know are you required to know it? I think I don't know if you're required to know it, but it would be, that's right still. I don't know if you're required to know the actual like chemical equation. It's nice to know it anyway. It's not that hard. You just need C6H12, no, sorry, other way. 6CO2 plus 6H2O gives C6H12O6 plus 6CO2 plus, oh well, and then there's light on the left side. Have there ever been questions asking for the chemical balance equation? I am unsure, but the thing is, yeah, well, bio don't learn balancing equations, but also there's I think it's okay for you to know, but it's not a requirement, so if you don't know it, don't stress type situation. I mean, the syllabus has changed also from when I did it, so I don't know whether or not you're like absolutely required. The textbook that I was using just said the word equation, so that's why I went with the word equation here. But yeah, exactly. So in, oh yeah, you do have photolysis that you do need to talk about, which I will talk about in a second. But In terms of SL content, I don't know if you need to know it. Sorry. I will search it up and I will let you know. But it's a bit of a like situation where you can know the word equation and that can be enough. Like you don't need to know the chemical formula of everything. If you know it, amazing, awesome, go for it. If you don't know it, don't stress. Okay. So, yeah, so carbon dioxide is what becomes yeah, exactly. So the syllabus doesn't mention the chemicals. It just mentions the word equation. So you can just know the word equation and it won't be an issue if you just know the word equation. But if you do know the chemical equation, it's not a bad thing to know in general. Is my opinion on it. Cuz I remember in the old syllabus, we needed to know it, but in the new syllabus, I don't know whether or not we do. Yeah, so I was talking about, yeah, so carbon dioxide is what becomes glucose in photosynthesis, and the way this is done is by adding hydrogen to carbon dioxide. And the way that happens is by photolysis, as Nemo said. Photolysis or photolysis, however you pronounce it, sorry, it's a bit of a struggle, is the splitting of water. So in the presence of light or energy in this situation, light energy, in the presence of light energy, water is split into its constituents, so hydrogen and oxygen, and that hydrogen therefore becomes gets added to the carbon dioxide in order to create glucose, and then the oxygen becomes a byproduct of photosynthesis, which is why when we say the word equation of photosynthesis, we add the oxygen at the end, so it's carbon dioxide plus water gives glucose plus oxygen. Yes? Good. Does that explain photolysis good enough? More or less. Yeah, no. So like I said already, you don't, in terms of the syllabus, in terms of requirements, you don't need to know the chemical equation. You only need to know the word equation for photosynthesis. There you go. Yeah, so I think everything after that is all HL stuff. So if SLs don't want to be here anymore, feel free. I won't be offended. It's just going to be a bunch of yap. Anything that you need to know, or if I'm missing anything from SL, let me know. I could be wrong because the SL, like the new syllabus, is very confusing still. If you use the chemical equation instead of word, is that okay? I think yes. I think it's okay if you use the chemical equation. I don't think they're gonna punish you for using the chemical equation as long as you get it right. I don't think they'll punish you. The slideshow from yesterday and the slideshow from today are all one slideshow, so I will put it up together after this session. Sorry, I wanted to finish both sessions before I add it in. Yeah, I'll do all the experiments later. So carbon dioxide enrichment stuff I'll also go through when I go through the experiment stuff. Because I also need to, because if I remember correctly, we didn't have that in our syllabus, so it's a new thing. So I want to do my due diligence and understand it before I try to explain it to you guys. So I'll go through it with the other experiments as well. But yeah, so that's everything for SL. Now everything I go through after this is all HL. Which is probably everything important about photosynthesis is in the HL syllabus. Everything that's not important is in the SL. And this is no offense to SLs, but yeah. Unfortunately, that's how IB is doing their syllabus now. Um, yeah, so are we all good? If there's any SLs that don't want to stick around, feel free to leave now. If you want to stick around and listen anyway, go ahead. Things about. Oh, so Sakura, you said your teacher covered a little bit of photosystems. As far as I know, it's not required in terms of SL syllabus. Um, it used to be last year. It's no longer a requirement. It's only a part of HL. So in terms of it won't be a question in the exam type situation. Um, but yeah. Okay, so HLs. This is gonna be really fun, or not fun, depending on how much you like bio. Okay, so there's these things called photosystems. Now, photosystems are basically molecular arrays of chlorophyll and other accessory pigments. So other accessory pigments can be carotenoid, etc., etc., with special chlorophylls as the reaction center, where at that reaction center is where the excited electrons. So, when we talked about absorption spectrum, and there's excited electrons, that basically electrons are excited by absorbing photons, and that happens because of the energy that's applied to it. So then the electrons move up an energy level. So when that happens in special chlorophylls, which is where the reaction central is, and then so pairs of excited electrons are emitted from special chlorophylls. Photosystems are located in thylakoid membranes. A thylakoid is a sac-like vesicle, which are present in different places. So in eukaryotes, which everyone should know what a eukaryote and prokaryote is. I'm not explaining that. In eukaryotes, in photosynthetic eukaryotes, so plants, etc., etc., thylakoids are flattened and arranged in stacks inside of chloroplasts. So they might also be called thylakoid stacks instead of just thylakoids. But yeah, so stacks of thylakoid are present in chloroplasts, and those thylakoid membranes. You can't be the one asking what a prokaryote is. That's crazy from you. Anyway, what was I saying? Sorry. Photosynthetic eukaryotes have thylakoids in their chloroplasts, and thylakoid membranes are where the photosystems are located. So there are two types of photosystems. There's photosystem 1 and photosystem 2, PS1, PS2, with different structures and functions. So PS1 is positioned where the thylakoid membrane is exposed to the surrounding stroma, whereas PS2 does not need to be in contact with the stroma. So does everyone know what stroma is? That's okay. People who are leaving, that's fine. Yeah, does everyone know what stroma is? Please define. Okay, so stroma is basically the easiest way to explain it is the cytoplasm of chloroplasts. So it's essentially, you know how mitochondria has matrix, and cells have cytoplasm. Chloroplasts have stroma. If that's a good enough explanation. So the photosystem 1 is positioned where the thylakoid membrane is exposed to the stroma so that it can accept, I've forgotten now, but I will explain it later on. But basically it has to be in where it's exposed to the stroma and PS2 does not need to be in contact with the stroma, so it's placed in different positions. Um, and then a fact apparently you need to know for some reason is cyanobacteria also have thylakoids with both photosystems, but they're not located in chloroplasts, and the pigments are arranged differently. Um, yes, that is true, B. You're right. Anyway, what was I talking about? Yeah, so cyanobacteria is basically a procar. I'm gonna be really careful with what I say. It is a prokaryote where the thylakoids are not present in chloroplasts, obviously, because they don't have chloroplasts specifically. So the thylakoids are still there. They still have both photosystems, but they're not located in chloroplasts, and the pigments will be arranged differently in cyanobacteria and other prokaryotes. But that's something you need to know, apparently. I don't know. It was a fact in the thing, so I just explained it. Another thing that I should talk about that I think I forgot to add in the slides. What's the next slide? Yeah, so I forgot to add this in the slides, but basically each photosystem has two types of functional units. There's a reaction center and there's an antenna. So the reaction center is what emits the excited electrons, and antenna complexes, I think is the right word, harvest the light energy and funnel it to the reaction center. So each photosystem has the reaction centers, which, like I explained, is special chlorophylls, and then the other chlorophyll and other accessory pigments are the antenna complexes, which absorb the basically absorb light and funnel it into the reaction center so that enough energy is generated for those excited electrons are what's it called, emitted from the reaction center, if that makes sense. Is that a good explanation of what the antenna and stuff is? Do you need me to go over it again? I can. No, I can. Sorry. It's just I got distracted while I was saying as well. Guys, if you want to have a conversation that's not related to this, please don't do it in chat, because I look at chat for questions and stuff, so I get distracted really easily. But yeah. No, you're fine. You're completely fine. But it's just a personal thing. Yeah, so I was explaining the structure of photosystems. So each photosystem has this thing called a reaction center and antenna complexes. Now, antenna complexes are basically what harvest light energy into the reaction center in order for things to happen in the reaction center. And then in the reaction center is where the electrons are then excited by the photons that are absorbed, and therefore the reaction center is where the pairs of excited electrons are emitted from. Antenna complexes have many different pigment molecules, not just chlorophyll, and these are positioned in a very precise arrangement. The advantage of having a large number of pigment molecules, it means that more photons of light are intercepted, and therefore the energy supplied is faster. So for example, like I said, it's not just chlorophyll, so it can be chlorophyll, carotene, beta carotene, there's xanthophyll, etc., etc. There's a lot of pigments that are present in this antenna complexes, because it allows for more energy to be harvested, and then that means that there's more energy provided to the reaction center, and faster even. PS2 happens before PS1, right? Yes, but I'm not going over that yet. I'm just talking about general photosystem structure. I will talk about that in literally a second. But I'm just going over the reasons why there's multiple pigment molecules in a photosystem, because apparently that's a learning outcome that you need to know. I don't know. I only just saw it. Sorry. I would have put a slide in, but I didn't realize that that was a requirement. But yeah, does that make sense? You need to know the advantages of having multiple pigments in the antenna complex. Because this wasn't a thing last year, but apparently we need to know that. You don't need to know, yeah, you don't need to know what exact extra pigments there are, but you need to know why extra pigments are useful and that they're located in the antenna complex and that the reaction center is special chlorophylls. That's what you need to know. Those are the 3 things you need to know. Okay, does that make sense? Okay, special chlorophylls are essentially just where electrons get the energy to be excited, and that's where essentially where the entire where the energy transfer happens, if that's a good enough definition. They're just specialized for the energy transfer to do, yeah, to for the electrons. Yeah, they provide special chlorophyll is where the light energy gets transferred into the electrons in order for them to be excited and therefore provide energy for the rest of photosynthesis. If that's a good enough definition. Yeah, perfect. Okay, so I think that's everything in this one. Let me just double check that I'm not missing anything. Okay, lovely. Now we're gonna talk about light dependent and then electron transport chain and light independent reactions. Okay, I have to remember all of this. So the way we're gonna look at this is from this direction and going up. I hope you can see my cursor, but basically we're looking at the bottom first and then we go around from left to right and up right to left. Light dependent reactions essentially means that obviously you need light, or the point is you need the light energy in order to convert it into chemical energy, and this is this part of photosynthesis is where light is required. After this, there's no requirement for light. But basically, so light, like I said, this is photosystem 2, this is photosystem 1. This is where, like I explained before, antenna complexes and reaction center exist, and therefore they release the 2 excited electrons. Now, when those 2 excited electrons are released, those 2 excited electrons, I'm gonna say this so many times, 2 excited electrons move from photosystem 2 to other membrane proteins and then through till photosystem 1. When this happens at photosystem, hold on, what was I saying? I'm like losing my train of thoughts. So at photosystem 1, more energy is provided through light energy, and the electrons keep getting pushed to a photosystem. This is an older picture, I think. I'll replace this picture with the newer picture. But basically what happens is when this is happening, NADP, not NAD. So yesterday we talked about NAD and FAD. Here it's NADP. So it's phosphorized NAD essentially. But NADP will become the receiver of the two electrons that are being pushed, and also hydrogen with hydrogen, them together form reduced NADP. So the two positive hydrogen radicals, I'll say, two positive hydrogens will get neutralized by the two electrons and therefore get reduce NADP to form reduced NADP. What makes the difference if it's phosphorized? I don't think you need to know that. I think you just need to know the fact that it's NADP and not NAD. Let me see if you need to know whether or not. I don't think you need to know it, really. It's just, you just need to know the fact that it's NADP and not NAD. Let me double check. No, not really. You just need to know that it's NADP instead of NAD. When it's converted to NADP, does it carry? Okay, so basically what happens is NADP exists in the stroma already, right? Now like I explained before, photosystem 1 has to be in contact with the stroma, and that's because NADP exists in the stroma. NADP plus hydrogen already exist in the stroma. Those two together accept the two electrons to form reduced NADP. NADP gets reduced by the hydrogen. So it's a reduction reaction, not a oxidation reaction. Sorry, if that makes sense. So when it's reduced, it goes from NADP to NADPH, or it's just called reduced NADP. So NAD and NADP are, in this situation, we consider them unrelated. NAD is something we use mostly in cell respiration. NADP is something we're using in the light-dependent reactions in photosynthesis. So you can look at NADP as an individual electron acceptor. So NADP becomes NADPH or reduced NADP, whichever way you want to write it. I think in the newer textbooks they're writing NADP. Yes. So reduction reactions are basically when you're either accepting a hydrogen or losing an oxygen. So the fact that you add a hydrogen, it becomes a reduction reaction. When you lose that hydrogen, it becomes an oxidation reaction. Sorry, I realized I didn't explain this yesterday, but that's what happens. So then NAD is reduced to add an H. NADP here is reduced to add an H. So you can either call it NADH or NADPH, or you can say reduced NAD and reduced NADP. I think in the newer textbooks they're trying to keep it to reduced NADP, but in our textbooks last year they added the H. So you can write it either way. I think both is correct. But essentially, yes. So by reducing it, we start beginning the other things. So I haven't finished explaining, I realized. So when I realized I didn't explain this area. So when there is at photosystem 2, when the two electrons are excited and start moving, for that excitation to happen, I don't know how to explain this, for them to be excited, there's also photolysis of water here. Now we talked about photolysis before, but photolysis in HL you have to talk about a little bit more. So essentially, special chlorophyll, which is P680, I think, in the textbook it says. Yeah, so absorption of light allows a special chlorophyll, P680, in the reaction center of PS2 to emit excited electrons. So that's where the reaction center is, essentially. As a consequence, this P680 is reduced and remains until it regains an electron. That electron comes from the photolysis. It is being recorded. Most of it anyway. I think I missed part of the beginning, but I will re-record that for people who missed it. Um, yeah, so the thing about photolysis is we talked about photolysis before, where hydrogen splits into hydrogen and oxygen, and then those hydrogens are used in order to make the glucose, right? That's the most basic part of photolysis. In HL, you need to know that um, the chlorophyll, as part of emitting excited electrons, gets reduced. So P680, that's the chlorophyll that you need to know, is reduced, and it will remain so until it gets the electron that's lost due to photolysis. So when photolysis happens, let me add this reaction to the chat. When photolysis happens, this reaction happens that I've just added into chat. So water turns into oxygen and hydrogen, and as a result of this splitting, there's also 4 electrons that are released. Now these electrons are what are accepted by the reduced chlorophyll in order for it to restart the process of the light dependent reactions all over again. So when in photosystem 2, when these 2 electrons leave, more electrons need to come back in order for this reaction to keep going. Those electrons come from this splitting of water into hydrogen. Does that make sense? I'll explain what happens to the hydrogen in a second. Give me a second. We're not there yet. I'm trying to see if people understand why photolysis is important here. No, it's fine. I will go through it again. So, basically what happens in light-dependent reactions, I realize I'm explaining this in a very scattered way, so I'll go through it all over again. So, at photosystem 2, photolysis of water happens where hydrogen and oxygen, where water splits into hydrogen and oxygen and 2 electrons. Now those 2 electrons are what is used by the reaction center in photosystem 2 in order to excite them. Those 2 excited electrons will then move across the thylakoid membrane into photosystem 1, and then is eventually accepted by NADP and hydrogen to create reduced NADP or NADPH. Because these 2 electrons will be moving, more electrons need to be accepted, which is why this photolysis continues happening. There's also the special chlorophyll in photosystem 2 is called P680. That's just a name you need to know. P680 is the special chlorophylls here that act as the reaction center. Now, while the electrons are moving across the membrane proteins, there's also channel protein pumps that pump hydrogen from the stroma into the thylakoid space, and obviously there's also hydrogen and oxygen from photolysis. This concentration of hydrogen rapidly increases as the electrons are moving across and later accepted. When there's a high enough concentration of hydrogen here, they move through, again, ATP synthase, where it moves the ATP synthase and therefore creates ATP from ADP. So this is similar to what happens in cell respiration, as I explained yesterday, and that will create ATP, which is then used in the Calvin cycle, etc., etc. Does that make sense? It all links together. It's all together. Okay, are we somewhat understanding how this works? Is there any questions that you have? Because I realize I just rattled through all of that really quickly. But if you have questions, ask them before I go on to the other stuff. But this is also, the light dependent reactions is also technically an ETC, like in cell respiration. So in cell respiration, it was electron transport chain. That still exists here. Is the H used to reduce NADP? It's both things. There's so there's hydrogen in the stroma. There's also hydrogen in the thylakoid space. The hydrogen that's in the thylakoid space, as a result of photolysis and also pumping from the channel proteins, those hydrogens are used for ATP synthesis, but then the hydrogen that already exists in the stroma is used to reduce NADP. So there's multiple functions of hydrogen within this. Sorry, that was a lot of yap. But yeah, I think Jess is writing a question, so I'll wait until they do. 2 hydrogen is pumped into the thylakoid space, but 2 hydrogen is also made due to photolysis. H2 and this H+. Yes, that is. So basically, yes, you're right. So there's hydrogen from the photolysis. There's hydrogen that's pumped because of the movement of the electrons. So then that creates a concentration gradient between the thylakoid space and the stroma, which then makes it go through the only available channel, which is ATP synthase. So yes, that's a good summary. I think that I have a summary on the next page as well. Yeah. So photolysis, like I have already explained, splitting of water, the 2 excited electrons from photosystem 2 is passed to a chain of carriers. Production of ATP is called okay, so this is something you need to know. So we said so then we talked about phosphorylation yesterday. In chloroplasts, this phosphorylation is called photophosphorylation because you need the light energy. The energy in phosphorylation in cell respiration is different than phosphorylation in photosynthesis because of where the energy is coming from. The energy in photosynthesis comes from the absorption of light, therefore it's called photophosphorylation rather than just phosphorylation. But at the end, again, the chain of carriers of the electrons are passed to photosystem 1, where then it's added to NADP to form NADPH, or reduced NADP. So the main 3 reactions that you need to know that happen here is the splitting of the hydrogen, the accepting of electrons by NADP, and the conversion of ADP into ATP. That's the 3 main things that happen in terms of reactions. Does that make sense? So I'm not going through what chemiosmosis is again, but basically it's the same thing that happens here. So it happens because like chemiosmosis in cell respiration and chemiosmosis in main product of the reaction in this situation is reduced NADP, ATP, and hydrogen ions. Those are the 3 main products. There's no singular main product because all of these are later used in the Calvin cycle, if I remember correctly. Not in the Calvin cycle, I mean in the synthesis, in the Calvin cycle, basically. That's harder to explain. I'll explain that later on. But there's no one main product in this situation. Because technically this is not the end. This is all processes so that the Calvin cycle and glucose can be created. Anywho, does this make sense? Somewhat. Yes. No. Maybe. Okay, perfect. Are there any questions before I go into... This is both PS1 and PS2. So the photolysis happens at PS2. The NADP thing happens at photosystem 1 / okay, let me find the picture that's in your textbook and let me add it to this thing. So this we weren't taught last year, but apparently you need to know this year for some reason, because the IB is getting more and more. Let me just make another slide. Hold on. Sorry, guys. Okay. So, there you go. This is at photosystem 1. So at photosystem 1, the 2 electrons that have been passed throughout the photosystem 2 and other channel proteins the electron gets deposited into NADP reductase, which is basically an enzyme that allows for the reduction of NADP. So in the textbook, I'm going to read out of the textbook right now, which is going to make me a really bad teacher, but I'm going to read it out anyway. So, using excited electrons emitted by the photosystem 1, reduced NADP is created. Each electron emitted is accepted by an electron carrier on the surface of the thylakoid membrane. The reduced electron carrier, NAD, binds to an enzyme, NADP reductase, also on the surface of the thylakoid membrane, and transferred the electron it is carrying to the enzyme. So, in order to explain what that said, this little square in dark gray, I don't know why all the pictures in this thing is just gray, but this dark gray is basically an electron carrier, also known as ferredoxin. This ferredoxin accepts an electron and deposits it to the NADP reductase. Now, it deposits both the electrons so that those 2 electrons can be used to reduce NADP into NADPH. And this happens at photosystem 1. Does that make sense? And then the things that happen at photosystem 2 are the photolysis, I was going to say hydrolysis, photolysis and the electron transfer starts at photosystem 2. But the reduction of NADP happens at photosystem 1. And then ATP is synthesized in ATP synthase, which is over here. Is that a good summary? More or less. Does that make more sense? Okay. Great. I will go through that later. Let's talk about light-independent reactions. I'll talk about thylakoids and stuff later. Let's talk about light-independent reactions. Okay, so there are 3 steps to the light-independent reactions. There's carboxylation, there's synthesis of carbohydrate, and there's regeneration of RUBP. Now, obviously, I'm going to go through that all in the what's it called? Calvin cycle. But basically, carboxylation is where you add carbon dioxide to RUBP or ribulose biphosphate. Now, ribulose biphosphate is essentially a 5-carbon sugar, which you might notice from ribose, which I think you should know in terms of carbs. Ribose is a 5-carbon sugar. So ribulose biphosphate is essentially a 5-carbon sugar, but it has 2 phosphates attached to it. Now, RUBP gets a carbon from this thing called rubisco. Now, does anyone know what rubisco is? I'm asking to be a good interactive teacher. What's rubisco, guys? That's fine. No, it's fine. That's good then, because you're learning now. Rubisco is essentially an enzyme which catalyzes this reaction. So the deposit of carbon dioxide to RUBP is done through a reaction which is catalyzed by rubisco. Unless T-Cell is about to correct me. I'm gonna wait. Oh, okay. Oh, it's fine. We just need to know Rubisco. It's a full form of something, but we just call it Rubisco. Okay, so it's basically a enzyme, like I was saying. It's an enzyme which catalyzes this carboxylation reaction, because otherwise this would be a really slow reaction and we need it to be a fast reaction. So, ribulose biphosphate, RUBP, plus carbon dioxide in the presence of Rubisco as a catalyst creates G3P, also known as glycerin 3-phosphate. Now, it forms 2 G3P actually. There you go. It forms 2 molecules of glycerin 3-phosphate. Now, light energy is not used in this. It's basically, so this process is called carboxylation, otherwise also known as carbon fixation, which is why you might know Rubisco in terms of enzyme when you talk about enzymes and stuff. So Rubisco is essentially responsible for carbon fixation. That's a like one of those star points, I guess. So this carboxylation creates G3P. Here you do not need light energy at all, therefore it's a light independent reaction. And Rubisco is present in the stroma of chloroplasts. And there's really, really high concentrations of it because carboxylation is slow as well. Fun fact is Rubisco is the most abundant enzyme on Earth, apparently, according to this textbook, which is very cool, which I did not know that happened, but that's cool. Okay, so, and then the next reaction after this carbon fixation is the synthesis of a carbohydrate, not the carbohydrate, of a carbohydrate. Now, glycerin 3-phosphate, or G3P, plus hydrogen atoms from NADP plus energy is what creates triose phosphate, or TP. So G3P becomes TP as a result of the hydrogen atoms that come from the previous light dependent reaction where NADP is reduced, and that reduction then deposits the hydrogen atoms in this reaction. So the NADPH comes, deposits the H, and then becomes NADP again, and it gets reused in the light dependent reaction. But this is where the hydrogen comes in. This is also where the ATP comes in, because energy is required in order for this conversion to happen. So then ATP becomes ADP again, because that breakdown will generate energy. Does that make sense? To the 5 of you that are here. Does that make sense? Okay, good. And then, since this is a cyclical reaction, yes, energy and NADPH are both required, which is why I said there's no main product of the light dependent reactions, because all of the products are reused in the light independent reactions. Yeah, so then, since it's a cyclical reaction, RUBP has to be regenerated. So the 2 glycerin 3-phosphate molecules create 6 triose phosphate molecules. Now, out of those 6 triose phosphate molecules, 5 of them, plus the supply of energy from ATP, gets reused to make RUBP. And then the 1/6 of the triose phosphate, or TP, is used to form several different types of carbohydrates. So it makes glucose phosphates or starch. Glucose phosphate is made by linking together two triose phosphates, so then it becomes, because each triose phosphate has three carbon compound, it's a 3-carbon compound. So 2 of them will make a 6-carbon compound, which is glucose. So glucose phosphate is created through 2 triose phosphates. And starch is formed in the stroma of plants by linking together many molecules of glucose phosphate together by condensation reactions. So this is how starch is made in plants, essentially. The triose, one of each Calvin cycle, one triose phosphate gets attached to make glucoses, and then those glucose phosphates are attached together to make starch. So, to show all of this in a really nice little graph, ribulose biphosphate, or RUBP, this is where carbon fixation happens using Rubisco. Carbon dioxide comes in. So there's 5 carbon compounds here, 1 carbon here. Those together become glycerate 3-phosphates, 2 glycerate 3-phosphates. So there's 3 carbons in each. And then ATP becomes ADP to provide energy. NADP becomes NADPH, or reduced NADP becomes NADP normal, where the H is deposited, and then energy plus the H make triose phosphate, and then 1/6 of that is used to make glucose phosphate, and then therefore starch. And the other 5/6 is used with some energy to make ribulose biphosphate again. I am going through photosynthesis. But yeah, so this entire thing is what's called the Calvin cycle. Does that make sense, guys? Yeah, I think that's everything. Is there anything else that I'm missing? Let me know. If there's any questions, let me know. I think that's everything. I could be missing things if they've added more. Why do I need all of that stuff? That's so real. That's so real. I don't know why. It's because it's also plants as well. So like, why do you need to know all of this? But I fear it's a requirement by the IB. I will put the slides up and the recording up later today when I get a chance. I will do that. I will do both. Promise. Because I think the first part of this is missing voice recording, so I'll fix that and then add the recording. Yes, most of the SL stuff is how's the stuff from light dependent transferred to light independent? Okay, let me go through that again. So, like I said, this is what happens in light dependent, right? Photolysis, reduced NADP, and ATP. Now, the photolysis is what provides the hydrogen. The photolysis is what provides the hydrogen for ATP to be created and also for the NADP to be created, right? So then NADP and ATP are both carried over here and there. So the NADP will deposit the hydrogens, ATP will provide the energy. So these two are transferred from the light dependent reactions into the light independent reactions. Is that good? Yeah. That's basically it. If there's more questions, I'll stick around for a couple minutes. Yeah, they basically, so all the light independent reactions happen in the stroma, and all the light dependent reactions happen in the thylakoid membrane / thylakoid space / and then it reaches the stroma, and then they float in the stroma until they do the light independent stuff, and then it forms starch. And then that starch gets carried around the plant and stuff like that for food and energy. Are all the light dependent reactions the Calvin cycle? Yes. So this is the main, that is what light independent reactions are. I just explained it in 2 different slides just so it's easier for me to explain everything and then show you visually, but that's essentially what the light independent reactions are. This part is the light independent reactions. It's called the Calvin cycle. No. So, this is what I was explaining. The Calvin cycle happens in the stroma of chloroplasts. PS2 is just where NADP is synthesized. Sorry, reduced. Let me go back. So, sorry, PS2 is where hydrolysis happens. Sorry, not NADP is synthesized. Wrong thing. Photosynthesis 2 is only where photolysis happens and where the electrons are provided for the light dependent reactions to happen. That's where the light dependent reactions happen. Calvin cycle is the light independent reactions, and that happens independent of the thylakoid. So that happens in the stroma. So that happens in the mirror of the, like, it's basically like the cytoplasm of chloroplasts. That's where it happens. PS1 is not where the Calvin cycle happens. So, let me go through this again. PS2 and PS1 are structures that are part of the thylakoid membrane. The thylakoid membrane is where the light dependent reactions happen. So where all of this entire cycle happens, where the electrons are moving, there's photolysis happening, there's hydrogen being pushed, ATP is synthesized. This is all the light dependent reactions. After this, NADP and ATP are floating around in the stroma, and that's where then the Calvin cycle happens. That's where Rubisco is, that's where RUBP is, and then that's where the Calvin cycle happens. So it's outside of the thylakoids. So it has nothing to do with the photosystems. The photosystems are only relevant to the light-dependent reactions. They're called photosystems for a reason. Photo means light. So that's where all the light-dependent reactions happen, because they're systems where the light is needed, if that's a better way to remember it. Does that make sense, B? So with light-dependent and independent reactions, the plant gets energy and sustains its life. Yes. Okay. So, in order, it doesn't get energy. It gets starch and glucose. That starch and glucose has to go through the Krebs cycle in order to be converted into energy. So it's interconnected. So it goes from photosynthesis, where it creates glucose and stuff, and then goes into cell respiration so that that starch and glucose can be broken down to form ATP, which can then be used, if that makes sense. So it's like, if you were looking at it linearly, photosynthesis happens, and then cell respiration happens, and then some of that ATP is then used here as well. But yeah. So, because this process doesn't generate any real ATP. It does, but that's used again. It creates starch, which can then be broken down in the Krebs cycle and stuff like that in order for ATP to be generated. Calvin cycle does not happen in the thylakoids. Oh my god, I'm gonna cry. Sorry. Okay, I don't know if I'm... No, you're fine. I don't know if I'm, like, complicating this too much. Calvin cycle happens in the stroma, and NADPH and ATP and the light-dependent reaction happens in the thylakoids. And yes, you're right that NADP and ATP is created to facilitate this Calvin cycle. That's completely right. The only thing that you got wrong in that sentence is Calvin cycle doesn't happen in the thylakoid. It happens in the stroma, which is basically the open space in chloroplasts. So all of this is happening in a chloroplast in a plant cell. Let me, hold on. So this is for example, this is a chloroplast, right? Now these are stacks of thylakoids, and the rest of the dot or like all the empty space is where the stroma is. So these stacks of thylakoids is where all the light dependent reactions are happening, and then outside here is where the light independent reactions in Calvin cycle is happening. Yes, you're right. Thylakoids is where NADPH and ATP is produced. And then outside here in the stroma, let me zoom into this picture. So like, hold on. Why are you being so rude? Oh my god. Entire screen. Okay. Okay. So, if this helps, this area outside is the stroma. These are the thylakoids, right? So now this membrane is where all the photosystem stuff happens. So on this side would be where photosystem 1 is, and on this side is where photosystem 2 will be, because photosystem 1 has to be exposed to the stroma and photosystem 1 does not. Photosystem 1 has to be exposed to the stroma and photosystem 2 does not have to be exposed to the stroma. So then the reason for that is the photosystem 1 is where NADP and stuff happens, so then it has to be exposed so that reduced NADP can be here. And then reduced NADP will be here, ATP will be here, and then the Calvin cycle happens in this region. I don't know if you can see my point, like my cursor. But yeah, does that make sense? Let me just... real. One part requires stroma and light contact, the other part doesn't. Okay, both parts require light, but one part doesn't require stroma contact. The reason for that is, let me go back to this diagram. Photosystem 2 does not require to be in contact with the stroma because it doesn't require any ingredients. The water that it requires is already present in the thylakoid space, right? The reason photosystem 1 needs exposure to the stroma is in order for the electrons to be accepted from the photosystem 1. So in this diagram, for example, this is the NADP reductase. This is the electron carrier, or ferredoxin. So then for this process to happen, it needs the NADP and the hydrogen, right? That NADP and hydrogen only come from the stroma. They're present in the stroma. They come from the stroma. Now, if it did have contact with the stroma, how would it do this part, right? The reason photosystem 2 doesn't need it is because there's nothing that it requires that would come from the stroma. Everything that it requires is already present in the thylakoid space, which is why it only needs that. Does that make more sense? Of course. Again, if you have more questions, I'll stick around. I'll stop recording now.